Hello and welcome to this webinar. My name is Lennart Eriksson and I will be the moderator and organizer of this webinar. This webinar is part of a guest presentation in Sartorius Stadium Data Analytics Solutions series of guest webinars and complementary webinars. The title of today's presentation is Directly from Chemical Structure to Human ADME and PK of Drug Candidates and Chemicals. And today we have the great honor of having Dr. Sven Hellberg and Associate Professor Urban Fagerholm as guest presenters. Dr. Sven Hellberg is a computational chemist and ex-manager in pharmaceutical R&D at AstraZeneca, and Sven is highly experienced with over 30 years of experience in QSAR modeling, compound design, and drug discovery. And his skills, experiences, and interests include molecular modeling, pharmacophores, docking, structure-based design, fragment evolution, hit finding, lead identification, lead optimization, chemometrics, and many more. Associate Professor Urban Fagerholm is also an ex-AstraZeneca person, a pharmacokineticist. Urban is holding an associate professorship in biopharmaceutics and pharmacokinetics at Uppsala University. He has a wide preclinical and clinical experience from more than 20 years in the pharmaceutical industry and academia, and has focused on prediction methodology, physiological models, mechanisms, and analysis and strategies in the ADME PK field. So with this introduction as a background, Sven and Urban, please, the word is yours.
Thank you, Lennart. This is Urban Fagerholm speaking. And hello, all listeners and viewers. So today we will present our company and what we're doing. We have a focus on in silico methods for prediction of human ADME PK. And we have an ambition to be a modern kind of company, try to reach as good results as possible. And one of the take-home messages today is that we actually developed methods that have beaten lab methods in accuracy, range, and cost efficiency. We will demonstrate some validation results and so. We're aiming for successful drug development, and we have a focus also to improve the safety and understanding of PK. And since we're working with in silico methods, we can actually also have a focus on reducing animal testing and also to protect the environment. The company was founded in 2014 by me and Sven. Since then we have developed 140 methods and provide data to more than 20 companies. We have a small biotechs and pharmacists that are customers of ours. We deliver to university research groups and also to tox institutes, and we have also delivered data to major international pharmaceutical companies. So next slide will demonstrate an outline of today's presentation. First we'd like to go through what ADME PK actually is and why it is important to predict that well. We present our prediction platform, how we built it and how it can be used, and we will also go through how we validated this platform, because that's very important to have a valid toolbox. We have compared our methods to established lab methods. We will demonstrate some examples how we have applied our models and what kind of output the customers receive from us. And we will end up with a summary. And we will welcome your questions, and we will respond to them in the end. So pharmacokinetics, that is actually what the body does to the drug. And that's an interplay between the drug molecule and the cells, more or less. So there's transport into cells, binding in cells, and transport out to cells, and also what happens in the cells. It could be binding, it can be metabolism. And the whole body is filled with different cells of different properties, and that actually will create a pharmacokinetic thing. So the major mechanisms: permeability, binding, distribution, metabolism, and excretion. gives us some major PK parameters that we're interested in. And I will demonstrate 4 of them that are perhaps the most important one, and that's a fraction absorbed. That means that how much of an oral dose that is absorbed from the GI tract. And the bioavailability is an important parameter, and that is how the degree to which the compound is entering the systemic circulation after oral administration. We have a parameter called clearance, that's elimination capacity of compounds, and the volume of distribution is a measurement of the distribution in the body. And combined, these actually gives us the exposure profile. You can see to the left or to the right that if you give a drug, it will be absorbed from the GI tract, it will enter the blood, it's distributed and eliminated, and the elimination phase is usually presented by a parameter called half-life. And when we have a PK prediction platform, we're actually able to predict also the dose and variability of compounds if we know pharmacologic data. So there are different types of PK assays, and in silico tools are usually used quite early in the drug discovery process, and further down this discovery and development line, more complex models are used. For example, in vitro tools, the Caco2 cell model and hepatocytes, microsomes. And before reaching human studies, more and more animal studies and data come in. And the general viewpoint seems to be that the more complex the model or the more animal-like, the better models are for predictions of human PK. That was demonstrated in studies 6 years ago, where it also was proposed that the animal in vitro methods were actually superior and should remain the golden standards for many years to come. And we also see combinations of different tools. So we have actually, when we start building this platform and this company, we dug deep into the data and to literature studies, and found that there are some compounds that are easier to predict and some compounds that are more difficult to predict. And when passive absorption, metabolic capacity, stability is intermediate and solubility is good and there's no excretion, it's usually quite, it's easier to predict the whole PK profile of a compound. But there are also some problematic scenarios. Compounds which are metabolically stable are out of reach for in vitro systems, and that means that you can actually cannot reach sufficient information to predict exposures in humans. You have some active components involved in absorption that could be problematic. Solubility issues are quite common. If you have excretion, strong binding to proteins, or phase 2 metabolism, it's usually problematic to measure and to predict it in humans. And if we take a look at in vitro studies in the literature, it seems that about 2/3 of compounds are out of reach for such systems, which is quite a lot. And the maximum error that we have found is actually close to 2,000-fold based upon in vitro data. So actually we have a need to find better ways to take care of these problematic compounds. There are quite many. And this slide demonstrates how oral bioavailability in different animal models and humans relate. And as you can see, there's not much correlation between them. Recently there was a paper in Nature that called for better methods to predict human exposures and safety. We have also found that the maximum prediction error based upon animal data is actually close to 6,000-fold. It was an anti-cancer drug that was given to humans, and they were overexposed by 6,000-fold. If this compound has been given orally, it would have been 1 million-fold overexposed, so that could actually have been linked to serious problems. We also found right recently that half a man was declared brain dead after receiving the first dose in the first time in clinical trials. So there is actually a need to have methods with lower maximum errors and better overall accuracy. So that's one of the grounds for our methods also.
So hi, this is Sven. So how did we develop our prediction platform? Well, it's in several steps. We go from chemical structure to human ADME PK. And the first step was to gather the data. We spent quite a lot of time on that. And after establishing the data, we started the machine learning and the data analysis. But we still needed a step after that to do fine-tuning of the parameters that came out of the machine learning. And then there's a step called PBPK modeling that Urban will talk about later on. But last, the most important step, I think, the validation. Can we really rely on these models to give accurate predictions? That was for us a very important step. But the first step was to gather the data. So this is actually where we spent the bulk of our time, almost 10 years on reading and checking hundreds and hundreds of publications of clinical data, quality controlling, sanity checking, and seeing if are the results reasonable and reliable, can we use them in a modeling sense. And once we had the data, we started to stratify the data according to mechanism of action. So we made subtables of data for compounds acting according to the same mechanism in the body. Once we had the data in place, we started to do the data analysis and the machine learning. And that was done during a 2-year period. So we developed QSAR models. The main methods used were principal components analysis, partial least squares, and support vector machines. So for those who don't know, quantitative structure-activity relationships, QSARs, that's a methodology where you collect a large number of compounds and describe the chemical properties with numbers accounting for various chemical properties of the compounds. And then you use PLS or support vector machine or other methods to find the relationships between the patterns in chemical properties, the relationship to the patterns in ADME PK properties.
So as I said in the beginning, we have developed more than 140 separate models for different PK mechanisms. And when we then take the smiles of compounds and predict each of these mechanisms, all data are translated to a PBPK model that we have built. And you see here that we have different boxes depending on different tissues and mechanisms. We have also spent quite a lot of time to re-evaluate all the equations and how things relate. So we have a different PBPK model than you usually see or what others have. And it seems that we've been able to reach a more balanced system without the skewness that you normally see. And as you see down in the middle, we have also a box for environments. We predict how much of a dose that is excreted out into the environment. I think that that is one of the unique parts of this system. And among these models, we have oral bioavailability is a parameter that is very difficult to predict. And if you go into the literature and try to find prediction methods for it, I think the only one with a lot of data is the one that you saw recently from animal to man. You saw the quality of that. We have also tried to find prediction methods for half-life, and we have not been able to find such models based upon in vitro data. So we've actually developed models here that that's been a great need for them actually. Fraction absorbed is one of the parameters that is behind or bioavailability, and it has been known for some time that efflux mechanisms, low solubility is a problem for lab methods, and we have focused on that, and it seems that we've been able to come up with quite good models, and combined with an in vivo solubility model that we developed, can actually predict compounds with very low solubility and also class them according to this bioclassing system. We have also focused on food interactions, several kinds of drug-drug interactions. We predict the mechanism and also the potential of them. Metabolic stability is an issue for metabolic stable compounds. So we have a model that can predict this parameter that we call a CLINT, or hepatic clearance. And the limit of quantification for our system is about 1,000-fold lower than you can find at an in vitro lab. So that's an important improvement. And we see that many customers have problems receiving the data that they need for these parameters. The total clearance, where we have included also the excretion mechanisms, is something that we have developed. We have methods also for blood-brain barrier uptake and CNS exposure. We have also focused on developing models for uptake into the body, not only the GI tract, but also via the eye, skin, and lung. You might be aware of this MIST initiative that you testing on metabolites, and we have tested the systems to predict also metabolic PK and exposure, also compared to the mother compound. And that is something that seems to be a quite unique opportunity for us and for the customers we have. We also some focus on prodrug PK, mainly the uptake process. And we also on some focus on the CYPs and binding in different organs. What we've done recently is that we took about 750 of the compounds that we have in a data bank, and we developed a PK space based upon that information we had. And as you can see in this part, we have been able to find that the major PK is actually determined by 3 major factors. And we have placed a compound called metoprolol in this space, and when we have a space, you can actually find the closest neighbor. So if a company comes with different candidate drugs, we can place them and see the lookalikes and the risk zones. You see here that this red zone is actually one risk zone for a certain drug interaction, and outside of that zone, there's no clinical information to demonstrate that you have a problem with. The next one here is that it's easier to follow a 2-dimensional plot than a 3-dimensional, so we've actually developed a 2D PK space as well. So this is an analog of the 3D space. It's easier to follow. And in this plot, we've actually tested how different kind of chemicals fit into this PK space, and these are the light blue dots, and I believe it's dark red ones also. And that means that our system can actually be used for other things than just drug molecules.
So the next step in our process was the validation. Validation is very, very important. It's a way for us to build confidence ourselves and to really believe in the performance of our models. And it's a way to build confidence for the customers. Can they rely on what we deliver? So the first step in the validation was to do an internal validation, doing a traditional cross-validation procedure. So we did that, and you see a plot here, an example of the observed oral absorption and the cross-validated predictions of absorption. we were very happy to see that in a predictive sense the models worked fine, so we could confirm that we had a predictive power. But of course, we wanted to do other ways, see other ways to look at the predictions. So we were thinking, can we do forward-looking validation? So check if we can use models based on sort of old drugs to predict the properties of new drugs, because that's sort of a more real life situation. We are going to use the models to predict potential new drugs that companies have. So we took a new take on the model system and developed model system only on old data from published up to year 2012. And then we predicted the recently published drugs from year 2012 to 2015. And we got about the same performance as for that validation as we got for the cross-validation. And so we could confirm that we can predict the ADME PK properties of new drugs based on the information we got from old drugs. But we still thought that we need to do even more validation. So the next step was to make a blinded validation. So we reached out to pharmaceutical companies and asked, do you have unpublished material? Do you have compounds that have gone into clinical trials where you have human ADME PK data that has not been published? And indeed we got positive feedback. So the companies sent us the compound structures and not the data. We used the compound structures to predict the ADME PK for the compounds and sent the predictions back to the companies. And then only after that we got the final verdict, so to say. Did it work? And as you can see from the plot there for the exposure in human after oral dose, the black dots are the cross-validated predictions, and the colored dots, the orange, green and blue, are from different companies, the blinded external predictions. The blinded predictions are just as good as the ones that we see from the cross-validation. So we were very happy with that. Now I think also from a methodological perspective, it's interesting to see that cross-validation really works. We could see here that what the indication we got from the cross-validation indicates very, very nicely the predicted performance that we could expect, that we got from the blinded validation. So we were very happy with this, and then we could move on and reach out to customers.
And one of the most important parameters in PK is actually the exposure after an oral dose, and that's actually a parameter, that's an integrated parameter based upon more than 25 underlying mechanisms. And there are some results in the literature where different animal in vitro methods have been tested and compared. And you see that the first four methods here are actually lab methods, and the data taken from the literature. And as you can see that this is the level that has been accepted to be sufficiently good for prediction of this parameter. And you can also see that the maximum errors are usually quite large, at least 100-fold. And for the in vitro methods, you have quite a lot of information that is lost. Compounds without information has been taken away. That means that we don't know the maximum error. So even for the best animal methods, you see that at least maybe 15% of all the compounds have not been able to quantify them, or they have not been included, or data have been excluded for some reason. In our validations for this parameter, we could actually reach results that were at least as good as the best of the lab methods. And we also reached 100% successful predictions. We didn't fail with any of them. So we say today that we are not better, but we're at least as good, and we have greater coverage and lower maximum error. So we thought that was a very important step forward for the company, for us. And in these external validation studies, we have sent results for 56 different compounds, and these customers, they validated them and sent back results, and we found that they could actually verify what we have claimed, which was very, very important for us, that the method seemed to work as well as we had hoped for. Another parameter is half-life, and we have not been able to find sufficient data in the literature to demonstrate how well lab methods work to predict this important parameter. But as you see here, we have been able to produce data that seem to be quite good. We have a median error of twofold, or at least maybe less than twofold. So this is just another example of some success of these methods. And in this case, we sent 75 results to companies, and the external validations were also similar to what we have done internally. So the model seems to work for this purpose as well. Usually, or quite often, we send data to customers just in a table, in Excel table form, so that they can analyze the data by themselves. But we also send reports, and when we send reports, we usually include reference data. And here's a table demonstrating what kind of information that we send to the customers. We have a set of parameters to the left and also the predicted information. And in this case, we have included diazepam, which is a compound that's quite similar to this compound number 4. And by comparing the predicted and observed data for diazepam, we can actually build confidence in the predictions. And that's the way we're working, to try to compare to reference compounds. We have also a parameter that we call prediction uncertainty index. That means that for every compound, we have an index that tells us how uncertain is this prediction. Is there some structural information or something that's very important to know that add to the uncertainty? And we have further developed the methods and see that we can actually reduce the errors by 30 to 40% by combining backup systems and to know about this uncertainty. I talked recently about chemicals, and we've developed models for uptake via the eye, the skin, and also the lungs, and we have validated those tools. You see the performance values below all the images. And this means that we can actually predict exposures, half-lives, and uptakes of various compounds, not only drugs. So here you see a figure about, we have food constituents, we have vitamins, we have different kind of drugs, we have natural compounds, and we have also colored these spots. And the meaning of that is that we have included how the degree to which the compounds are excreted out into the environment. So the red dots are actually those who have an extensive excretion out into the environment. You see DDT is included here. We predicted a half-life of about 6 months, and that's close to the true value. And this slide demonstrates a set of cosmetics, how they are absorbed across the human skin. This is the dermal permeability model that we have, and The numbers might not be so important, but the rank ordering gives more information. So we have actually used these models for various companies to predict uptake, and also today we have a new method that predicts the dermal bioavailability. And we believe that this is an important step forward because we can actually replace animal studies with just computing.
Okay, to summarize, when we look at quality of predicting ADME PK man, looking at quality, speed and cost, it's no surprise that the in silico methods, the data analytical methods, are speedy and cost efficient. So that's what we guessed already from the start. But what it took even us by surprise was to see that when we look at quality, the in silico based estimates are just as good or even better than the ones that you get from lab methods. Definitely better than what you get from cell in vitro systems, and better or on par with the best lab animal methods. So when we look at quality, speed and cost, we come out looking very good, I think. And then to look at another factor, that's the range of applicability. Maybe you remember that 2/3 of this pie chart was red when we looked at the in vitro systems, but as Urban mentioned, we generally can give nice predictions for all drug-like compounds or chemicals in the same range. So we think this is a very important aspect, that you can always get a reasonable answer when you use the in silico methods.
I would like to add something, and that's about the range of molecular weight here is about 150 to 750. That's our safe zone. We've also looked at larger molecules up to 1,300 in molecular weight to extrapolate data, and the result seems to be almost as good as in this safe zone. So we looked at the peptides, and so up to 1,300 in molecular weight. It seems to work well.
So to summarize, it's interesting to see that high quality in silico models have very nice properties. When it comes to quality, you can, in the ADME field here, you can get a complete range of human ADME PK parameters. They outperform or are on par with the best in vitro and in vivo methods. They can be validated, and you can see the predicted performance. Cost, significantly reduced lab costs, and very short delivery timelines. And last but not least, the ethical part. Reduced animal testing, improved safety. As Urban mentioned, the maximum errors are significantly lower with the in silico methods. And we also have an environmental aspect of this. And this concludes our part of the presentation, and we hand the word over to Lennart.
Sven and Urban, thank you very much for this extremely interesting presentation. Crystal clear to my mind, and it is one of the most impressive validation scheme I have seen. There are already a couple of questions typed in the question field. We will just stop the recording and shortly be back. So for the moment, thank you so much for your time and attention.